I'm a fellow in vascular neurology. Pretty much what that means is that I'm still in training, but I'm hoping to specialize in stroke. And so this is my lecture. Thank you all for coming today. So how do we learn new things? How does our brain heal after something like our stroke? And how come some patients after a stroke heal better than others? Can your brain really change? These are interesting questions, right? So welcome to my lecture. Today we're going to talk a little bit about neuroplasticity. So the brain is fascinating. It allows us to form memories, express emotions, coordinate movement, speak, and it ultimately makes up who we are. Yet there's still so much about the brain that we don't know. Our brain is constantly changing, and what we thought we knew about the brain is constantly changing as well. There used to be a lot of misconceptions about the brain and about how it works. For example, it was once believed that the brain did not change after childhood, that whatever we learned was hardwired and fixed in place by the time we became adults. And the only thing that did change was maladaptive or negative, for example, in a stroke. And it was once believed that once your brain was damaged, it could not be reversed. That is, your brain could not learn new information. But we now know that is simply not true. And in fact, your brain can and does adapt after damage. And every time you learn an information, you learn a new skill or refine a new movement, you're in fact changing your brain. And this change continues throughout our lifetime. This ability to change, to adapt, to modify, and to learn is called neuroplasticity. And that's what we're going to talk about today. So how does this happen? How do we learn? Well, there are three main ways that neuroplasticity takes place. It can happen on a chemical level. This involves signaling between brain cells called neurons. And this signaling leads to downstream actions that lead to actions and more reactions that ultimately lead to learning. For example, the short-term learning of a new skill. Over time, with enough changes, the brain itself can be altered. Do you remember, for example, when you were young, how to ride a bike? At first, it was really difficult. You may have forgot where to put your feet, how to stop, how to steer, and you may have fallen off your bike multiple times. But with enough time and enough practice, this became second nature. And now for most of us, even though it's been many years since we last rode a bike, you can do without much difficulty. And this happens over time because with continued chemical changes, the structure of the brain can be altered. That is, a short-term memory becomes a long-term memory. And we can see this in other cases as well. The very first recorded demonstration of neuroplasticity was by Italian anatomist Michele Vincenzo in 1793. Vincenzo described experiments in which he paired animals, trained one extensively for years, and then dissected both. He discovered that the cerebellums of the trained animals were substantially larger. Richard Davison, a neuroscientist at the University of Washington, has led experiments with the Dalai Lama on the effects of meditation on the brain. He found that long-term or even short-term practice of meditation was associated with altered activity and density of structures in the brain associated with attention, emotional regulation, and the ability of the body to heal itself. Take individuals who are born deaf. Their auditory cortex and other hearing association areas can undergo what's called compensatory plasticity. The auditory cortex, which is usually reserved for processing auditory information in hearing individuals, is now redirected to serve other functions, especially for vision, somatosensation, and compared to hearing individuals, deaf individuals actually have enhanced peripheral vision, more effective visual search, and faster response times to visual stimuli. And so this brings us to the last way your brain can change, and that happens by altering its function. As you continue to use a region of the brain, that region of the brain becomes more excitable. And the more you use it, the more excitable it becomes. More excitement leads to increased function and increased ability for you to regulate that particular part of your brain. And this is occurring all over your brain all the time. Yes, in certain areas, it may not be occurring all at once, but in concert, they're occurring together. So if you take all these changes together, chemical, structural, and functional, these are the changes that make up neuroplasticity and the ability to form new learning. So let's look at this in a bit more detail. So when we think about neuroplasticity, we need to go back to the very basic level, neurogenesis, or the ability to develop new neurons. As we briefly touched on before, it was previously believed that the brain was a non-renewing organ, that it could not regenerate. And yes, for the most part, this is true. Once you damage your brain, these areas cannot grow back, at least not in the same way, for example, if you were to get a cut, how your skin can regenerate. There are some areas of your brain, in fact, that are constantly regenerating. For example, the olfactory centers and the hippocampus, which is involved in learning. You ever wonder why certain smells ever trigger such an emotional, nostalgic response? Well, that's one of the reasons why. However, for other parts of your brain, it's not so easy. These areas don't regenerate, like your hippocampus. But these areas can and do regenerate in other ways. And that's by altering the chemical structure and function of the neurons in the region around the damage. So how do neurons do this? How do neurons influence each other? Neurons influence each other by altering their synaptic connections. So what does this mean? So each neuron is made up of a cellular body surrounded by antennae, which you can see here, and they're called dendrites. And then attached to each body of a neuron is called an axon. And this arm-like extension extends to the cellular body or the dendrites of another neuron. In between the two neurons is a small gap called the synapse. And this synapse allows small molecules called neurotransmitters to be released. The neurotransmitters, in turn, bind to receptors of the second neuron and change how the other neuron operates. They can only do this if there's enough stimulus. So it's all about the reception. So if a neuron receives a weak stimulus, and this is kind of just a general overview of a synapse here, then only a small amount of neurotransmitter can be released into that cleft, that synapse, to communicate with the second neuron. If there's only a small amount of neurotransmitter that's released, then the second neuron cannot wake up and become active. Therefore, the stimulus is no longer able to propagate and activate other neurons. So whatever signal that was triggered initially stops. It cannot propagate further. It cannot activate any other neurons. And these signals cannot be sustained. However, if a neuron sees a strong enough stimulus and in high enough frequency, then it can release a large amount of neurotransmitter into the synapse. With a large amount of neurotransmitter, the second neuron can now become active or depolarize. Over time, with enough activation, each signal is generated for a prolonged amount of time, and the neuron can undergo gene transcription and the creation of new synapses or new connections between neurons called Synapogenesis. With more synapses surrounding a neuron, more neurotransmitters can be released, and the more activation that can occur, making the connection between neurons even stronger and faster. This is called long-term potentiation. Similarly, if there's not enough strong stimuli or not enough strong stimuli and high enough frequency, the connections between neurons becomes weaker and slower. This is called long-term depression. Over time, long-term potentiation and long-term depression leads to both physical and functional changes in your brain. So remember, the more you use it, the more you use certain areas of the brain, or the more stimulus you receive, certain parts of your brain will grow larger and become more active and easier to use. In concert, other parts of your brain become less active and less easy to use. So that's great, right? I just told you that your brain was very neuroplastic. But if our brains are so neuroplastic, then why is learning so difficult? Why do some people learn easier than others? Why do we forget things? And why don't people recover fully after a stroke? That is, what really controls and limits our neuroplasticity? In truth, this is a very difficult question to answer. There are many factors, including baseline genetics, age, underlying structural and functional potential, and environmental factors that control our neuroplasticity. There is no one-size-fits-all approach to learning and to maximizing your neuroplasticity. Neuroplasticity is an individual as a fingerprint. What may work well for one may not work well for another. And what may come easy to one person may not be so easy for another and require much more practice, more repetition. So things like underlying genetics, age, we can't change those things. But what can we change? How can we maximize our potential? How can we maximize our neuroplasticity? So let's talk about neuroplasticity in the context of stroke. So the good news is that overall, stroke survival is improving. In the United States, stroke went from the third leading cause of death to the fourth leading cause of death in the early 2000s. That's great news, right? But in reality, all this really means is that we are better at keeping people alive after a stroke. And as it turns out, more and more young people are getting strokes each year. The burden of disability after a stroke is actually increasing. And it's increasing for longer periods of time as people are living longer with their disability. In fact, stroke is the #1 cause of long-term disability in the world. So what can we do? How can we minimize disability and simultaneously improve the quality of life after a stroke? What studies have been done on neuroplasticity in stroke? What is the evidence? So let's first talk about what happens at a stroke. We know that a stroke occurs because there's a disruption of blood or oxygen supply to the brain. But after injury, after that area of the brain that is damaged, there are several changes that occur in the brain, and most of them occur almost immediately. Some are involved in preserving brain architecture and function so that further injury cannot take place. Some are involved in healing and the development of new signaling, while some are involved in the creation of new function through Synapogenesis, and some are all maladaptive and can lead to further injury and brain dysfunction. As it turns out, ischemia is a strong inducer of gene expression in the brain. Evidence has shown that more than 90 different genes can be induced after a stroke. And all these genes have different effects and different functions on the brain. The study of these genes, as well as the molecular studies, have led to the development of novel interventions that promote post-stroke rehabilitation neuroplasticity, which I'll continue to talk about next. So let's start by looking at what's called neurotrophic factors, or the study of growth factors, hormones, and its association with neuroplasticity and rehabilitation after a stroke. So the brain is filled with a dozen of different neurotrophic factors. And these factors are involved in cell signaling and cell regulation. These factors have been studied widely in different disorders, especially in stroke. For example, one of the factors is called brain-derived neurotrophic factor, and has been reported to improve memory and motor function, and also to reduce atrophy in pyramidal neurons after injury. And this is not only true for brain-derived neurotrophic factor, but also for dozens of other factors in the brain, including nerve-derived factor, insulin-like growth factor, glial cell-derived neurotrophic factor, and even erythropoietin, which you may have heard of. However, most of these studies looking at neurotrophic factors are in the early stages. They're limited to preclinical trials or to studies in animal models. These factors, unfortunately, they don't act the same way in everyone, or even the same stage in each person. In fact, the effect of one factor in one person can lead to recovery at one stage, and that same factor can lead to injury and maladaptive responses in a later stage, even in the same person or in a different person. So I mentioned erythropoietin. Erythropoietin, one of the largest trials of erythropoietin was conducted to date, showed that compared to placebo, erythropoietin was associated with increased mortality in patients with acute ischemic stroke, even though in rat models, it was shown to improve neurogenesis and regeneration. So currently, unfortunately, the benefits and utility of neurotrophic factors is still unknown. We need to do more studies to determine the impact of these factors and how they can affect our brains in learning and healing after a stroke. So what about pharmacological therapies? You've heard of probably many of them, and in fact, may have read of many of them. So unfortunately, currently, there are no drugs approved by the FDA to improve recovery after stroke. But there are a lot of evidence for drugs that can help. And these here are listed drugs that can help with the strongest of evidence. So SSRIs, dopaminergic neurons, noradrenergic drugs, cholinergic drugs, amphetamines, and statins. So I'll first talk about SSRIs. So SSRIs is abbreviation for serotonin reuptake inhibitor. And it allows the serotonin, pretty much what it does is it allows serotonin to be more available in the brain. SSRIs are widely used in anxiety and depression, but over the past few years, it has also been used in recovery for stroke. Previous studies with SSRIs shows that it can help reduce inflammation, increase neurogenesis, facilitate cortical excitation and inhibition, and improve post-stroke depression. One of the first landmark trials using SSRIs was the FLAME study, which stood for fluoxetine for motor recovery after acute ischemic stroke. This was published in The Lancet in 2011. This was a double-blinded, placebo-controlled trial that randomized patients with predominantly a motor stroke with hemiplegia to either 3 months of 20 milligrams of fluoxetine or to matching placebo. Patients randomized to fluoxetine had improved motor scores at 90 days. And overall, this study was pretty small, but it did show initial promising results. Unfortunately, despite this initial promising study, several other studies following this study did not show similar efficacy. So based on these studies, it's really unclear at this point if SSRIs have a clear benefit for motor recovery. But that being said, in certain cases, we do use this to help patients recover from a stroke. So dopaminergic drugs. So dopamine is known as a happy neurotransmitter. It's involved in reward, learning, and plasticity. It plays a strong role in creating neuroplasticity by enhancing neuron excitability. It enhances synaptic transmission, protein trafficking, gene transcription, and motor skill learning. A randomized double-blind placebo-controlled trial was published in The Lancet in 2001 that studied 53 patients within 6 months of a stroke and found that patients taking 100 milligrams of L-DOPA per day, also known as Synapse, same drug that's used in Alzheimer's, combined with physical therapy, had better motor outcome than patients who did physical therapy alone. And again, this is another example of initial promising trial. However, there's been several more smaller studies that have been done on dopamine and stroke recovery. And unfortunately, the same results and benefit on neuroplasticity was not the same. And this may be secondary to large variability in environmental, cognitive, and psychological factors for these patients during this study. What needs to be done now, unfortunately, like the SSRIs, is a little more studies. So about other drugs, I mentioned noradrenergic, cholinergic, and amphetamines. So noradrenergic drugs are drugs that sort of act on your flight or fight response. Drugs like you may have heard of clonidine, which is used in blood pressure, and these drugs have been studied in post-stroke recovery. Drugs like clonidine have been shown to improve neuron excitability, increase neuronal activity, improve arousal and executive functioning. But to date, unfortunately, there have only been a few trials that have studied these, and most of them are small, non-randomized, and have variable results. So the results for these is really inconclusive. The same thing goes for cholinergic drugs. Cholinergic drugs that have been used, for example, in Alzheimer's, have been published in inconclusive results. Amphetamines, you may have heard these drugs like Ritalin, Adderall. These are used to improve attention and focus, especially in children and adults who have ADHD. There have been limited studies in stroke with amphetamines, but the initial studies have shown that it has improved some neurotransmission. However, to date, again, these trials are really, really small. They're non-randomized. And depending on which trial you look at, the effects are not the same. So unfortunately, for most drugs, the studies are inconclusive. So statins. So statins not only improve lipid metabolism after a stroke, and pretty much it's now the mainstay of therapy, especially moderate to high intensity statins like Lipitor, which you may or may not be on or have a family member that's been on. So statins not only improve lipid metabolism by lowering LDL levels that can contribute to a stroke, but can actually improve plasticity and functional outcome by modulating endothelial function, process, attenuating inflammatory and oxidative stress damage, and improving angiogenesis. So currently we recommend, even in patients with good LDL levels or LDL levels that are less than 70, to remain on a statin therapy unless they have significant side effects or contraindications to statin therapy, because you do, even if you have normal LDL levels, get that benefit, get that improvement with neuroplasticity. So why are drug trials lacking? Why are studies so variable? Well, it's kind of a complex question, and it doesn't really have a clear answer. There are many factors that can contribute to post-stroke recovery in terms of efficacy of medications. But one of the major factors is time. As with many diseases, stroke evolves over time, and biological targets shift. There is not a designated therapeutic window for which certain drugs can be used for maximum efficacy at this time. Drugs that promote recovery 1 week actually may be harmful for the brain in the next. But timing is really just one aspect. Underlying biological mechanisms, genetic variability, stroke size and distribution, post-stroke therapy, psychiatric factors, and coexisting medical conditions all play a role in stroke recovery. That is, stroke is really heterogeneous. Each person is truly unique. Just like there is no single therapy for all patients with pneumonia or cancer, there is no single drug that is useful for enhancing recovery across all patients in stroke. So you say, I've read about biomarkers. What about biomarkers? Are there biomarkers that can give us information on how patients recover after a stroke? After all, we do use certain biomarkers in other diseases, take plasma RNA levels in patients who have HIV, or brain natriuretic peptide in patients who have heart failure. Is there something like that for stroke? Well, yes and no. And this is sort of one of the frontiers of the next research for neuroplasticity. Yes, there are several biomarkers that can be studied in stroke, both to predict risk of stroke, but also assess the ability to recover. However, none to date have been significantly shown to be efficacious across the board. So why is that? Well, in order to use a biomarker, you need to have an accurate one that's very little variability when it comes to timing and to individuals after a stroke. A good biomarker must be in all the pathways of the disease process and must fully capture the net effect of the treatment on the clinical outcome. And so, as I mentioned before, the biomarker research is just beginning, and a lot of centers are looking into this currently. Unfortunately, right now, maybe in the future, we'll have one that we can test and that we can measure, but additional studies are needed. So what about novel therapies? More recently in literature and on the news, we've heard about stem cells. So what are stem cells? And how can they aid in neuroplasticity? So stem cells are undifferentiated or partially differentiated cells with the ability to form different cell types. Take, for example, neural stem cells. These are one type of adult stem cell that is found in the brains of humans and animals, and it has the ability to form three major central nervous system cellular types: neurons, which we talked about, astrocytes, and oligodendrocytes. And studies in neurostem cells, or stem cells in general, are just really beginning. In the early 2000s, researchers from the National Institute of Health arranged a meeting to develop clear recommendations for the research and development of stem cells in the treatment of stroke. It was from here that several studies were launched. One of the first reported clinical trials using stem cells was conducted in 2000. 12 patients with basal ganglion infarcts were transplanted with human neuronal cells stereotactically. In a follow-up period, after 6 months, 6 out of 11 of the patients showed increased activity on positron emission tomography or PET scanning and had improvement in memory, stroke impact scales, and a non-significant improvement in functional outcome. Since then, several more studies have been conducted, some with mild or non-significant benefits, others with no benefit, and increased risk for adverse events like seizures, unfortunately, tumor formation, bleeding. Today, studies in stem cells are ongoing. So currently at KU, we're actually participating in a trial using stem cell therapies. This is a relatively new study that came rise in the past few years. It's called MASTERS-2. So MASTERS-2 is a randomized, double-blind, placebo-controlled trial examining the effectiveness of allogenic adult stem cells in patients with acute ischemic stroke. Initial data for the trial was published in 2016 and show that patients who received the multi-stem, is what they call the stem cell in this particular trial, had faster recovery, shorter hospital stays, shorter ICU stays, lower rates of mortality, life-threatening adverse events, as well as greater proportion of patients receiving excellent outcome as measured by modified Rankin scales or motor skills at 90 days. So this is kind of sort of really promising. And we're still at this time conducting, enrolling patients in this particular study. So what about other therapies, sort of novel therapies that you've heard of? Some people may have read about hyperbaric oxygen therapy. So what is hyperbaric oxygen therapy? Hyperbaric oxygen therapy is relatively non-invasive therapy performed in a pressurized chamber where pure oxygen is administered at above atmospheric pressures. Hyperbaric oxygen therapy is quite beneficial in certain conditions such as carbon monoxide poisoning, burns, and infections. And its benefit in stroke and TBI studies is just beginning, and it can show benefit by reducing edema or brain swelling, inflammation, cellular death, and improving cellular perfusion. However, unfortunately to date, as I mentioned earlier, there's only a handful of randomized clinical trials involving hyperbaric oxygen therapy in the treatment of stroke. And all these results have unfortunately been inconclusive. Most of these trials are small, non-randomized, and unfortunately poorly conducted, with the results that are not statistically significant. Wide variations in treatment protocols, stage of patient enrollment, inhibit really the ability to draw conclusions across trials and form protocols for treatment. And then furthermore, unfortunately, hyperbaric oxygen therapy, like drug therapy, benefit depends really on the timing. And you may get particular benefit in a particular window during a stroke, but outside that window can cause significant damage because it leads to cellular damage through formation of reactive oxygen species. Thus, currently the data on hyperbaric oxygen therapy is inconclusive, and unfortunately more studies are needed before we can say, it's significant benefit. So what about technology? How can we use technology to maximize our neuroplasticity? Well, the most well-known and probably the most well-studied technologies for the treatment of stroke involved invasive brain neurostimulation. And the two most common studied ones is transcranial magnetic stimulation, or TMS, or transcranial direct current stimulation, or DCS. DCS has been around probably since the 1800s, and TMS has been around since about 1985. Most of the time, both these studies are actually used and most studied well in depression. For the treatment of depression, it has very good efficacy. In both techniques, an electrical current is applied to the scalp. The electrical current then can stimulate cortical neurons either directly via direct current stimulation or indirectly via transcranial magnetic stimulation. These in turn help modulate cortical circuit excitability and augment neuroplasticity. Both techniques have been shown to improve function after a stroke, including improved motor function, gait recovery, aphasia, neglect, treatment of neuroplastic pain, and mood disturbance. However, as I have mentioned earlier, and you'll see me continue to sort of harp on this issue, is that most of the studies with non-invasive brain stimulation are quite variable, have large inter-individual variability, and it makes standard protocols difficult to develop. Therefore, the evidence that shows the effectiveness of these techniques is unfortunately still ongoing, but it's something that's currently being well studied. So along the lines of using technology to help the brain recover is the use of prosthesis in the recovery of a stroke. So we as a medical society have become comfortable with the use of prosthesis implanted in various parts of the body. For example, a pacemaker in the heart to prevent an abnormal arrhythmia, or an artificial knee to help increase mobility. Now, over the past 4 years, the idea of prostheses have reached the brain. Prostheses can be used to input information into the brain. For example, if we are blind, it is now possible to have a small camera inserted into your glasses, and images sent from that camera get sent to a small chip that's set on the back of your retina. This chip then stimulates the eye like a regular image would to process images. And what this does, it allows people that are profoundly deficient in sight to see. They can see some light, some shadows, and maybe even general shapes, so they can better navigate the world a little bit easier. Prosthesis can actually now be used to output information from the brain, as in the case of a stroke. The picture of this woman you see here is a 50-year-old woman who was affected by a brain stem stroke that left her paralyzed for almost 15 years. In this particular study, researchers used a tiny electrode, it was about the size of a fingernail, and planted it on the surface of the brain. The signals from the electrode are then transmitted from the brain to a computer to move a robotic arm. So what she is doing in this picture, in fact, what she has just done, is she used her brain signal to move the robotic arm, to pick up a bottle of coffee and bring it to her mouth to drink. This is the first time that she's been able to do this independently in 15 years. So this is remarkable, right? And so the studies in prosthesis are just currently expanding, and they're expanding at an exponential rate. Prostheses are now used simultaneously with acute rehabilitation therapy in the recovery of stroke by helping the brain and the body coordinate, aid, and execute movements. That is, prostheses can help the brain maximize its neuroplasticity. So we learned some pretty unique things, some interesting therapies, right? But the truth is that the single most important therapy, the treatment with the most and best evidence to maximize your neuroplasticity, is acute rehabilitation through physical therapy, occupational therapy, and speech therapy. There is no magical pill, no magical lab test, biomarker, a mechanical device that can better aid in your recovery than acute rehabilitation and long-term therapy. For patients who undergo acute rehabilitation, compared to those who don't, have lower rates of mortality, decreased rates of hospital readmission, higher likelihood of returning home, both in the short and long term, and have greater improvement in functional outcome long term. The timing and frequency of rehabilitation is also important. Rehabilitation, when permitted, should begin within 48 hours of stroke onset, and often much sooner. One of the largest studies in post-stroke rehabilitation was the AVERT study. This study was published in the journal Neurology in 2016 and showed that patients who started therapies within 24 hours of onset of their stroke with the goal of out-of-bed activity had significant odds reduction of achieving a functional outcome at 3 months following a stroke. So remember earlier when we talked about neuroplastic changes after a stroke, they occur almost immediately. So this is why it's important to start therapies very early. It's also important to continue therapies. Studies have shown that stroke recovery can occur years after the initial injury. It was initially believed that stroke recovery was really, limited to a few months, but this is really not true. It can happen many, many years after recovery and actually throughout your whole lifetime. Patients who continue therapies had greater improvements in functional outcome despite the amount of time it has been since the onset of a stroke. And we have many studies to show this. Remember, in order for your neurons to make synapses and to continue signaling, you need to have a strong enough signal and a high enough frequency for that signal to continue. If not, it will, it won't propagate. Your neurons will no longer fire. So yes, rehabilitation is not easy, but studies actually show that the more difficult the rehab is, the actually better it is for your brain. And it's the difficult, repetitive nature of rehab that makes recovery possible. The bottom line is you have to put in the work. So I do want to mention that, neuroplasticity, I told you many good things about neuroplasticity, but it can also be negative too. It can work in both ways. It can be positive. You learn something new or you refine a new skill. It can also be negative. Neuroplasticity can lead to depression, addiction, chronic pain, and even loss of a skill. For example, studies have shown that people who are constantly exposed to negative stimuli have disruption in their underlying neurochemical balance, leading to increasing depression, suicidality, and increased ability to cope with stress, and even increased ability to heal. Over time, if this negative stimuli is not stopped or corrected, the effects on the brain may actually be permanent. So it's very important to recognize this fact during stroke recovery. Remember, everything you do and everything you don't do affects your brain. Just as neuroplasticity can improve your brain after a stroke, it can also hurt your brain as well. So what have we learned today? Well, first, we have learned that your brain is tremendously dynamic. It's shaped chemically, structurally, and functionally by everything you do, and also by everything you don't do. Secondly, there's not a one-size-fits-all approach to learning, and not a one-size-fits-all approach to recovery. Neuroplasticity is variable. For some of us, it's going to take a lot more practice to learn something than it is for others. And that's okay, but repetition and individualized learning approach is key. Lastly, and most importantly, we are in the driver's seat for our learning. It is up to us to maximize our neuroplasticity. There is no magical pill, no magical biomarker or treatment that can enhance our neuroplasticity. The only way we can increase our neuroplasticity and learning potential is through hard work and repetition. So thank you all for coming to my lecture today. I hope that when you leave here that your brains have all changed for the better. And if you have any questions, I'll open up